Our next speaker is Dr. Steve Goodman, who is an MD epidemiologist and statistician who founded and directed, is it SPORE?
Yep.
Okay, SPORE, the Stanford program on research rigor and reproducibility. And he also co-directs Metrics, the Meta Research Innovation Center at Stanford. His scholarship and teaching is in the foundations of inference and clinical research methods. He is interested in how institutional change can improve research informativeness. He is also an associate dean in the Stanford School of Medicine and a member of the National Academy of Medicine. And so today, Professor Goodman is going to be speaking on the 4 Ds of veridical biomedical science, which we have above. Oh, design, 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 and data. So introduce our speaker. Thank you.
Thanks so much. It's really delightful to be here today, and I'm learning a lot with every passing minute. And hopefully this talk will expand that conversation. So I want to take a step back, and as you can tell from the title of the talk, I'm going to explore, in a sense, the epistemologies, the current epistemologies of medical research, how we think about evidence, and what I've learned about VDS and the PCS framework, which I will say, and I'm heartened that Binyu said that she had to go look up Veridical. I did too, and I learned that it meant truthful. Which, and I have to say to add the word truthful to something that was previously called a science, that takes a bit of chutzpah, or to use a sort of sexist metaphor, sort of ballsy to say, we have science. But we want to modify it with truthful, because I thought that's what science was. But of course, she's absolutely onto something, which is why many of us are worried about reproducibility and all the things that she talks about. So I'm listening. Dr. Kornblith actually was a great start to what I'm going to be talking about, which is, I've served much of my career serving as a sort of interlocutor between the worlds of medicine and the worlds of data. And I put up here listing, it is not just my CV, it is my conflicts of interest. I've been firmly embedded in the world of medical research for many, many years. I started actually studying math, and I've been around long enough, and I heard you don't actually want to hear from those of us who've been around a long time, but you invited me, so you don't have a choice. I've been around long enough, so when I applied to medical school, I didn't get into many because they literally told me several that you can be interested. I told them I wanted to unite math and medicine. And they told me you can be interested in math or you can be interested in people, but you can't be interested in both. So that's why I took off time during medical school to go back to study math because they made it so inhospitable to me. But eventually I did find my way back to a world that united all of them, and I became a pediatrician, like Dr. Kornblith, and have been in the worlds of math and medicine and biostatistics and epidemiology for my whole career. And these are just all the different pools worlds I've been trying to straddle. But I've been mainly facing the medical world, taking what I've learned from in the what we'll call it the data analysis world, and teaching medical scientists how to best use data. Now, I started to learn about veridical data science, obviously with Dr. Yu's paper on this topic. And I will say, just going back here, I actually thought I was doing truthful data-intensive science most of my career until I read about this, so I realized I could do a lot better. But hopefully this will be a bidirectional conversation. So this is the data science life cycle, and you've all seen it, and at the core of it is stability. Now, I just want to say, before I say anything else, that I am absolutely in favor of truth. I'm not against truth, and I'm not against methods that move us towards more truthful statements. In fact, I'm on record as talking about this. This is a column in Nature, the Worldview column, which should be retitled "Fantastic Ideas That Don't Go Anywhere." And so here was my a few years ago. I said at the end of every published article, the investigator should say what degree of confidence they have in their findings, just on a scale of 0 to 100, or just the probability that it's true. I don't think it's going anywhere. I would encourage you to do it, or urge your collaborators to do it. And if you succeed, please send me an email. I'll use it in my next presentation. But I think that much of what we're talking about gets to this. That is, how do we faithfully represent the uncertainty we have in findings? And actually, we should start with our own. And so I think the proper representation and measurement of uncertainty is absolutely core to this. Now, this is the updated version of the data science life cycle from the book. And I read this with tremendous interest because I don't think I have it. Do I have a laser pointer here? Oh, yeah. And I'll talk about a number of these, but 3/4 of my career, I noticed, isn't here. It falls in this gap. That's actually what I'm going to be talking about. So it doesn't appear here. And that's part of that is design. And I'm going to show you the figure I've used for almost 30 years in teaching about about research and inference. And this is how I organize it. And actually, this represents the very traditional way that biomedical scientists think about things. First of all, we start with a scientific question or conceptual framework, which is a relationship between concepts. We then go to the study question, which is a relationship between measurements in an experimental system. Sometimes we call them the specific aims. We then operationalize that through a study design, how measurements will be made and organized to permit valid inferences, then study conduct, study analysis, and then study reporting, interpretation, and limitations. And I don't have a circle here, but I do have a arrow back, which I call induction, where we go back and revise our initial beliefs. And to help compare the two, I've organized them here, and you see some interesting differences in terms of where we focus the granularity. So up here, there's a lot more granularity, and it doesn't map. The study design doesn't map to a clear concept here. Data collection, I call study conduct, but data collection for me is a much more unstructured activity than gathering it in the context of a designed study. Then we have, so there's a lot more granularity here than here, but what I call study analysis, we have a lot more granularity here. And a lot of steps which I sometimes go through with students, but I think are underappreciated as sources of variability and uncertainty, which is, of course, some of the focus of PCS. Here we end up in the same place, and here I'll say we end up in the same place. So I'm going to explore what both epistemologies can learn from each other through these differences. So, R.A. Fisher was one of the first people to write a popular book on the design of experiments. It was called, very cleverly, The Design of Experiments, 1935. And this is one quote from him a few years later that emphasized his focus on design. And probably most of you have heard this. To consult the statistician after experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. We repeat that a lot. And what he's really saying is, obviously, you have the data in hand after the experiment, but what you haven't done is design the experiment. So this is a statement about design. He was saying that you cannot resuscitate a data set and get the same validity if you haven't participated in the design. One thing that's unique about this display is I think it's almost the only picture you'll ever see of R.A. Fisher smiling. Now I just want to show you how deeply embedded design issues are in the epistemology of medical evidence. So this is just a classic, what we call the levels of evidence pyramid. And what you see is in this one, it's stratified totally by design. RCTs, cohort studies, case control, case series, and case reports. And now down at the bottom, we have expert opinion. And here they've added a little thing at the top they distinguish between filtered evidence and unfiltered, systematic reviews, meta-analysis. They unbelievably put practice guidelines at the top. I'm not going to comment on this, but just the point that they stratified only by knowing the design, nothing else. I actually find this to be hysterical because at the bottom is expert opinion. Who do we think put together these pyramids? So there's a bit of circularity there. So this has been refined, fortunately, in light of current knowledge. And here we have another version of the pyramid adding in animal and in vitro research. And this is the grade evidence rating, which is an improved evidence quality metric, which incorporates, to some extent, the ability to adjust for confounding, imperfections in the RCT. You see it starts with an randomized trial still at the top. Observational study starts at low, and you can promote these or demote these based on other characteristics having to do with conduct or finding. So it's a little bit of an advance. And then we get this with this is the Robbins Eye tool, which is based on the fidelity to target trial emulation to an RCT, and this is particularly a quality tool for interventions. So now we're getting this is from 2016, now we're getting some refinement, but it's still based on design. It's still based on design. So design is at the heart of the assessment of medical evidence. And it's so much a part of it. This is the table of contents from the Annals of Internal Medicine, where I've actually worked for over 30 years. I guess that dates me, huh? And this is just from last month. And what you'll see, the subhead for every single study is the design of the study. That is, you cannot begin to interpret the study before you know something about the design first. Then if you go and look at one of the abstracts, more than 50% of the abstract is about the design. So again, this is part of the DNA of medical research, is you start with the question, but the assessment of the truthfulness, the uncertainty is based, number one, on the design, and then you flow into the conduct and analysis. Now, I'll go through one example here of a group that's been trying to look at the fidelity, the ability to copy RCT results with observational data. And this is out of Harvard, and I'm going to start with some graphics they worked on and I'm not going to actually step through what these represent. They represent different design features of observational studies to allow you to see if the design is close to that of either to each other or to RCTs. And I'm just going to flip through these and then show you the actual paper I'm interested in showing. So I'll just say what we have here are time is on the bottom line. And here we have the timing of various assessments of the covariates, of the entry into the cohort, of the time when people took a medication, of when you, when you excluded people, when you had the information that allowed you to exclude people. So you want to know why people are in your cohort. And here just representations of different designs. And again, up here we have exposure-based cohort entry restricted to adherent users. And you can see the pattern of colors. And again, I'm not going to go through it, but you will see graphically how different these complex designs are. Now we have a visualizing a nested case control design with risk set sampling. This is all based on health, information extracted from claims data or health records. And they make this point that there's two differences from what we'll call ground truth. One is from emulation differences, which is the difference between the real world evidence, that's the RWE, the observational evidence, and the RCT. And they have a whole bunch of categories. I'm told I only have 1 minute. I'll take a couple more, but there'll be time for questions. Okay. And then there are the bias differences, which are typically taken care of analytically. But we cannot take care of the design differences, for the most part, analytically. We have to recognize them. They're embedded in the data set, and they're embedded in the questions that we're asking. Every design answers a different question. So sometimes if it's a poor design, it's answering such a vague question that we don't know what the answer means. Now, this is the example I wanted to show. I don't know how many of you are familiar with it. It's been going on for a long time. It's RCT Duplicate. And it was commissioned by the FDA, by the same group at Harvard, led by Sebastian Schneeweiss, Jesse Franklin, and Shirley Wang. And what they did is they took 32 clinical trials and they tried to emulate them using databases. And these are some of the design emulation challenges that I just mentioned. I'm not going to go through every list, but they went through, they had the parameters, so they had sort of a ground truth. They had the parameters of the RCT, and then they used the observational database to see how close they could come to that design. This is what a grid they made, everything in blue. So across the top are all the design parameters. Going down on the left side is just all the trials. And in blue is everything that was good that they were able to come close to for that trial. And in yellow are the emulation challenges. And that's all the time we have to orient you to this. But just trust me, this is all about design. But these are the results. So for the half that were best emulated, and they were able to assess this before they looked at the results. So this is, again, only based on design. Only based on design. You see that here's the RCT effect coefficient, and here's the emulation coefficient. A very tight clustering with just a few outliers, and I could talk about those, but I'm not going to, along the line of identity. Among those that were poorly emulated, again, which they predicted ahead of time, tremendous scatter. Huge amount of scatter. So this was all based on design parameters without looking at the data. Just looking at the design. So let's contrast this with a project that was led, I think, by Russ Poldrak, where they didn't necessarily have a ground truth. This is one of my favorite papers. I think it's just absolutely fantastic, which shows a problem when you have less constraint by design and perhaps less causal restrictions, that is, less causal knowledge. And what happened was, and we really should have Russ come here and do it. I'm only going to do it in like 30 seconds. It was part of the neuroimaging analysis replication study. The actual study was 108 people with fMRIs performing one of two versions of a task. They had 9 hypotheses about which brain region was involved in the task feature, and it was given to 70 experienced fMRI teams with a 100-day deadline. So what did they find? Well, here are the parameters of the analytic pipeline, and no 2 teams of the 70 used identical analytic pipelines. No 2. This is getting exactly at the point that Dr. Yu was talking about before. Okay, did they come up with different results? Well, they absolutely did. So here, this is the number of the hypothesis. This is the percentage of the 70 that came up with a significant result. And you might say there's a lot of consensus here. 84% of them did, but 16% didn't. Here, 94% did, and so there was a lot of unanimity here, but a lot of disagreement in the middle out of the 70 teams. And this is actually my favorite part, and Russ, you get a huge amount of credit. They asked them, how much confidence do you have in the findings? So this, by the way, puts the lie to my previous piece about how important this is. And how confident are you that it was similar to the others? So this is fantastic. What you see is basically it runs between 6 and 8, no matter what. No matter how much disagreement there was, they had pretty high confidence in their findings and pretty high confidence that it was similar to the others. Complete lack of appreciation of the variability induced by the analytic pipeline. So these are sort of generic list of data science challenges. I would say that we have to have a discussion about how the PCS framework and data science in general deals with issues of design, data quality, and measurement. I think what we're seeing today and what I look forward to learning more about is how much better we can do in terms of uncertainty quantification, propagation, and model validation. I think this adds a lot to that. The area of causal inference, I'm not so sure about. That is actually the main goal of most medical studies at their core. Even if it's not obvious, the causal inference is at the root of our confidence that we can use, that we can generalize results. Because if it's not based on causal factors, we find that they fall apart in other contexts. So generalizability and causal foundation are two sides of the same coin. So this is the last slide. I think there are mutual lessons here, and I would love to hear more conversation about it today. I still think that design and conduct trump analysis. I don't think you can make the evidence stronger, even with fancy. The design puts a ceiling on how strong the inference can be. The strength and truth value of a claim cannot be stronger than the design. But the design isn't always articulated, and that's a problem. So analytic strategies and data sources either follow designs or they imply designs. And if you learn how to look at the code and the analytic strategy, you can figure out what design they're assuming that they are following, and that tells you the strength of the evidence. Where causal understanding is weak, that is, unknown confounders, missing data, and many of the issues that we confront in medical research and in epidemiology, I think the impact of model flexibility, data cleaning, all the things that Bin was talking about, is almost always underappreciated and underestimated. I think this is a huge problem, and it's a huge problem in my field. We just fit models and we report them. It's unbelievable. We don't have the awareness of these multi-analyst studies that we're increasingly doing. So I think that the robustness and stability checks through the VDS, DS, the life cycle can produce truer uncertainty estimates than those typically reported. But I think exploration of how we use the multiple results you get is really an issue. A lot of times in epidemiology, we do sensitivity analyses, but we don't incorporate the range of estimates from those uncertainty analyses into the expression of uncertainty. We simply say our conclusion isn't changed, instead of saying the uncertainty interval is larger. And finally, we, or maybe it's just me, need to understand better how the empirical predictiveness and stability relates to the role of design in causal inference and in uncertainty measurements. And I'll just leave it there. So thanks so much.
Thank you very much for most informative, insightful talks. And your point's completely well taken by the design. It's probably reflecting my own ignorance of the design because I haven't worked on it very much myself. Hopefully the next version will do better. And in terms of causal inference, I do think that we have one paper. I think the stability, when even you have randomized trials, for the adjustment, people have the same analysis pipeline choices again. So one thing I was hoping to do, if you use different adjustments for causal inference, you have this uncertainty right there. And I think stability is closely related to the invariance in causality, right? A lot of things you can put transfer learning, different environment. So I think there's a close connection. In the cardiology project, we really try to do, I believe, predictability and stability work towards a little closer. It's not the proof to causality. So that's something I think we can copy into observational studies.
Yeah.
So definitely we should continue this conversation.
That's why I put up causality here and stability there. Again, but I think we need to understand better what the connection is between those.
Some version of my other talk, I do have this to discuss more, but it's excellent that we started this conversation. Thank you.
Thank you.
One more question.
So this is more a comment. So I think perhaps that the design, can come into the place within the PCS framework. Like when one comes with this sort of the perturbed data. So I think that in the perturbation, simply instead of doing just the bootstrapping, you have to one should consider that the study design and how to decide that the creating the perturbed data. Perhaps that's the one easy place that the study design can come in. Of course, it doesn't address that whether that from the general point of view that the data collected is addressing the question that they address. That bigger question perhaps it cannot be addressed, but I think that within the framework of the PCS, perhaps that the design can come in there.
It can, but it doesn't address things like how did that subject even come into the study? Why were they measured? Were there symptoms generating the measurement? What's the quality of the training of the person who's doing the measurement? I mean, there's a whole series of questions that have to be asked that are embedded in design and conduct that are not in the data. And that's where the design comes in. So you're absolutely right. I think around the edges, there are, again, you're sort of talking about implied designs that isn't explicitly laid out. So sometimes you can't figure out what the design was from the data. And then you have, I think it's hopeless. I actually think it's hopeless. because you have to know why people were there and why things were measured. And you need to have some sense of causal direction, and those are key to bias. Then some of this bias cannot be adjusted out if you don't know the structure of the data. So I'll just say that. So there are lots of things we do with imagined designs, and sometimes the designs are so simple that maybe we don't really need it. But when you talk about some of the complicated things that were being studied in the examples I showed, you can't do it without design. But in some problems, you absolutely can. Particularly if you're just talking about pure prediction and you have no interest in causal factors. Sure. I can't say no to you.
So one thing, can you speculate, how do we do the design for large language model development?
That's so when the very people who are developing them say that they don't completely understand how they're working, what I would say is, so it's a form of large scale, what we call, in the philosophers called not large scale naive inductionism, done on such a scale that we never appreciated its power. It's, as we know, that's a lot of it is probabilistic. So if the very developers of the technology can't tell us 100% how it's working, and it's just based on complex correlational structures, I'm not going to pretend from this podium to be able to tell you what the design is other than complex correlational structures.
Thank you.
And let's thank all of our speakers.